DNA is organized in a very specific way within your cells. You often hear DNA referred to as a double helix, which just means it has two strands. It's made of two strands that are connected together, and that it's a twisted molecule, kind of like this one, right? Two strands connected together, and it's twisted. But DNA, right, is two long strands of these monomers or building blocks called nucleotides. And a nucleotide has three parts that make it up. It has a phosphate group, it has a deoxyribose sugar, that's where deoxyribonucleic acid comes from, and it has a nitrogenous base. It may have one of four nitrogenous bases attached to it. It might have adenine, thymine, cytosine, or guanine. So any of these nitrogenous bases plus a deoxyribose sugar plus a phosphate, that's one nucleotide. And our cells, each one of our cells has trillions of nucleotides to make up our genome. These two strands are held together by hydrogen bonds, which allow for that durable structure. However, they're also weak enough to be easily broken for DNA replication or transcription. In eukaryotes, the DNA can be found in the nucleus in its double helix form. However, it's not all stringing out everywhere. To help it stay organized, it's wound around these proteins called histones. So these big green boxes represent histones, which are actually groups of proteins. So our DNA double helix is wound around these in order to form nucleosomes. Right? A nucleosome is just a length of DNA that's coiled around a core of histones. This is important. It's going to help our DNA keep from getting all tangled around, and also it's part of what lets us turn DNA off and on. These histones, when they are, or nucleosomes, when they are loosely near one another, all right, they're going to form Euchromatin. All right, so this is DNA in a cell that's active. These genes are turned on. They're being transcribed. Their proteins are being made. So, for instance, all the cells, or all the DNA that makes skin in my skin cells and makes it all of its skinny magicness, are on. They're on there in the Euchromatin form, all right, in which DNA is loosely coiled around those proteins and it's transcribable, right? Conversely, the genes in my cells that are turned off are in a form called heterochromatin, all right? And so these are the genes that are turned off. So again, in my skin cells, the genes for producing bone, the genes for producing fingernails, the genes for producing liver cells are turned off. They're in the heterochromatin form. So in heterochromatin, right, our DNA is coiled tightly around the histones and it cannot be transcribed. Those genes are turned off. The method by which these are actually attached tightly and wound together is when methyl groups, these little tags, these methyl tags, will attach the DNA chemically to the histones, turning that DNA off. Now, at the beginning of mitosis, right, in the cell cycle, all your DNA starts to coil tightly together and further condense until it's all in heterochromatin form, and we can actually physically now see under a microscope the chromosomes themselves. They become visible, right? So DNA double helix winding around our histone proteins, our nucleosomes coming together, coiling tighter, condensing more, and that's when you see that characteristic sort of X-looking thing. Really, it should look kind of more like that, because you have one of these and it replicated, and you have one of these. So since this is after the S phase, after DNA replication, right, there are now two copies of the chromosome. So that's one copy, that's another, right? That's one copy, that's another, and they're attached together. so when you're looking at that chromosome, when you can actually see it under the microscope, you're going to be able to see that only during mitosis of the cell cycle. And what you're actually looking at when you see that X-shaped chromosome are two, they're called sister chromatids. So one copy of the chromosome is one sister chromatid, and the other copy is the other chromatid. So they are identical copies of DNA, and they're held together in the center at a special place called a centromere. Now the centromere is a place, so it's not exactly like a separate thing from the chromosomes. It's a place, a locus or location on the chromosomes where this group of proteins called the kinetochore attaches. So, right, we have our long strings of DNA, they're coiled together. There's a special location where they meet and are held together by these proteins called kinetochore proteins. So they are important that they are at the centromere because this is where our spindle fibers are going to attach and help us to align those chromosomes and then separate them during the mitosis portion of the cell cycle.